I'm not sure where to put this since it's random, so I'm just going to talk about it with my purine and pyrimidine lectures. So you classify hormones based on their chemical structure or what kind of receptor they bind to. So 3 structures of hormones are peptides, steroids, and amino acid derivatives. Peptide hormones are from the hypothalamus, anterior pituitary, posterior pituitary, pancreas, and GI tract. Steroid hormones are coming from the adrenal cortex, the gonads. Amino acid derivative are coming from, so our thyroid hormones are made from amino acids, and our catecholamines from the adrenal medulla are made from amino acids, tyrosine. We also have hormones that bind to plasma membrane receptors on the surface of a cell and receptors that are intracellular. So I remember steroid and thyroid, they have an I, so that stands for the intracellular. And these bind to the what are they called? The elements on DNA. Like they're always like, this would be thyroid. It's like TRE. I forgot. Whatever. Plasma membrane receptors are, so peptide hormones bind to receptors on the cell surface, and so do catecholamines. And I remember this because catecholamines like norepinephrine and epinephrine bind to adrenergic receptors. And I remember those being on the outside. And then I remember steroid hormones being intracellular because I remember always being taught that steroids are lipids, so they just diffuse through the bilayer. Then peptide hormones, I just think of them being large and needing to bind on the outside of the cell. And then energy storage hormone is insulin. Insulin's in the fed state. It promotes the storage of glucose and tags. Energy utilization hormones are the ones that are occurring during like fasting and starvation. They're trying to get stored nutrients out into the blood. So glucagon, cortisol, norepinephrine and epinephrine, and growth hormone. Growth hormone is secreted during fasting and exercise. Now I'm going to talk about the last unit, which was purines and pyrimidines. So purines and pyrimidines are part of DNA and RNA. Purines are pure as gold. They are double ring structures that consist of adenine and guanine, so that's why I say as gold. Pure for purine, A for adenine, G for guanine. Both of these are found in both DNA and RNA. Pyrimidines are single ring, so I think of pyrimidines being a purine but cut in half. So I remember cut as cytosine, uracil, and thymine. Cytosines found in DNA and RNA, thymines only in DNA, and uracils only in RNA, and I remember the R in uracil for RNA. We also have sugars in our DNA and in RNA structures, deoxyribose and ribose sugars, and the only difference is deoxy means without oxygen. So on carbon 2, there's just a hydrogen. A ribose sugar has an oxygen on carbon 2. So that's the difference. So deoxyribose sugars are used in DNA. Ribose sugars are used in RNA. A nucleoside is one of these bases plus one of these sugars. So when you take purines like adenine and guanine and you add a sugar to them, adenine becomes adenosine, guanine becomes guanosine. So they turn from -ine to -sine. Pyrimidines, when you add a sugar to cytosine, it becomes a nucleoside. It becomes cytidine. When you add a sugar to uracil, it becomes uridine. When you add a sugar to thiamine, it becomes thymidine. So pyrimidine nucleotides become dine. The nucleotides are phosphate, sugar, and base. So the difference from nucleoside to nucleotide is the phosphate. You can have 1, 2, or 3, and that'll determine whether it's a monophosphate, diphosphate, or triphosphate. So that brings me down here. So when you're naming them, the first letter is the base. The second letter is the number of phosphates, mono, di, or tri. And the D signifies that it's the DNA form, so you add deoxy. So this would be, for example, ATP really stands for adenosine triphosphate. As we said before, adenine, guanine, and cytosine are all seen in RNA and DNA. Thiamine's only seen in DNA, so you'll only see it with D nucleotides. Uracil is only in RNA, but uracil is used to make thiamine, and in the process, you make these 2 DNA nucleotides for uracil, but they're not functional. They're only produced in the pathway, but we don't put them in DNA. So base plus sugar, nucleoside. When you add a base, sugar, and a phosphate, you get a nucleotide. You can also say that as nucleoside plus phosphate is nucleotide. When you add a bunch of nucleotides together, you get a nucleic acid. And nucleic acids are DNA or RNA. There's a lot happening with nucleic acid metabolism, but essentially you eat another animal's meat. We eat their DNA, which sounds kind of gross, and we turn their dietary nucleoproteins into nucleic acids. And I remember that because proteins turn into amino acids, so nucleoproteins turn into nucleic acids in the stomach. In the small intestine, nucleic acids get further broken down into their building block nucleotides. We remove a phosphate from a nucleotide to get a nucleoside, because this is the only type we can absorb. Once we absorb it into our intestine, we send it to the liver, where a few things can be done. We can add the phosphate and get a nucleotide right away again. We can remove the sugar from a nucleoside and then just get a free purine base. We can just excrete the purine base and break it down into uric acid. Or we can add a sugar and phosphate back onto the base to make a nucleotide, and that's known as salvage. The same thing can be done with pyrimidines. If we have a pyrimidine nucleoside, you can add the phosphate right back and get a pyrimidine nucleotide. You can remove the sugar and get a free pyrimidine base. We can break the free pyrimidine base down even further into beta-alanine or beta-aminoisobutyrate, or we can save them and take a free base, add a sugar and a phosphate, and make a pyrimidine nucleotide again. but let's focus on purines for a second. So purines, we saw our double ring structure, pure as gold, so adenine and guanine. They can be in DNA and RNA, so you can use a ribose or deoxyribose sugar, and you can use 1 phosphate, 2, or 3. Purine rings are made up from a bunch of different substances. So I heard this mnemonic: 9 pure hearty ass grabbings. 9 is B9 in the form of N10 formal tetrahydrofolate, which is giving a carbon to the ring. Pure is for knowing that we're talking about purines. Hearty is knowing HCO3 is giving a carbon, bicarbonate. As is for aspartate, knowing that aspartate's giving a nitrogen. Glycine, knowing that it's giving nitrogen and carbons. And glutamine, knowing that it's giving, there's 2, so the 1G is for glycine, 1G is for glutamine, but there's 2 glutamines. Basically all the amino acids, like the importance is their amino group, so they're all donating a nitrogen. When it comes to purine synthesis, we can make them from scratch, which is what de novo means. We can do phosphoribosylation of the free base, so adding a phosphate and a ribose, so sugar, onto the base to get a nucleotide, or you take a nucleoside that has the base and the sugar and then just add a phosphate through phosphorylation. So the process is you do the pentose phosphate pathway, starting with G6P from glycolysis and turning it into ribose sugar, ribose 5-phosphate. You take PRPP synthase and turn ribose 5-phosphate into PRPP. Then you take PRPP aminotransferase and turn PRPP into IMP. IMP can go one of 2 ways. It can make adenine nucleotides or guanine nucleotides. So IMP dehydrogenase turns IMP into XMP. GMP synthetase turns XMP into GMP. Add a phosphate, you get GTP. Add another, or GDP, you add another phosphate, you get GTP. And then here, on both sides, ribonucleotide reductase is turning the RNA diphosphate nucleotide into the DNA nucleotides. You can go this way. IMP turns into adenylosuccinate using adenylosuccinate synthetase, or adenylosuccinate breaks up into adenylosuccinate lyase cuts adenylosuccinate into AMP and fumarate. Add a phosphate onto AMP, you get ADP. Add another, you get ATP. What determines so this pathway is stimulated and inhibited by different things. So PRPP synthase is stimulated by ribose 5-phosphate because you need the sugar to do the pathway. It's going to be inhibited by all of the nucleotides produced, because this is the product of this pathway. So if we have the product, we don't need to make any more nucleotides. PRP is the rate-limiting substance. PRPP aminotransferase will be stimulated by PRPP because that's what it's working on, but it'll be inhibited by IMP and then the rest of the nucleotides, which makes sense because if we have the nucleotides, if we have purine nucleotides, we don't need to be doing this purine nucleotide synthesis pathway. We can save energy and use our energy somewhere else. GMP production. So if we have high AMP, we'll that'll stimulate the production of GMP, because we always want our A's and G production to be equal. So like GTP and ATP will inhibit these enzymes again, because they're the product of the pathway. Von Gierk's disease is when you have no G6P. So you can't turn G6P back into glucose. If you can't do that, you can't let glucose out into the blood, so you'll have hypoglycemia. If you have hypoglycemia, then you'll need to get energy from somewhere else, so you'll have lipids going into the blood, and that'll cause hyperlipidemia. And if that goes even further, you'll produce ketones for energy. You'll also have hyperuricemia, because if you can't turn glucose 6-phosphate back into glucose, you're forced to go this way and produce purines. So if you keep going this way and producing purines, the more purines you produce, the more you have to break down. And we know that when you break down purines, you form uric acid. So the more uric acid will be formed. And that's why you get hyperuricemia. Purine catabolism happens when we're breaking down purines into uric acid, like I just mentioned. So we start with IMP, we break it down into GMP and AMP, the purine nucleotides, remove a phosphate and you get the purine nucleoside. Remove the sugar from guanosine and you get guanine. This is the free base. You have adenosine, and you use this molecule ADA to turn it into inosine. If you inhibit ADA, you'll get this disorder that I learned in immunology, severe combined immunodeficiency, which is when you're born without lymphoid tissue, and you don't have B and T cells, so then you'll be prone to pretty much all infections. That's the Bubble Boy. So Bubble Boy had SCID and he needed ADA accommodations. That's how I remember that. And then PNP turns inosine into hypoxanthine. Xanthine is the common product of hypoxanthine and guanine. Xanthine oxidase turns hypoxanthine into xanthine, and it turns xanthine into uric acid. If you inhibit xanthine oxidase, you won't produce uric acid, and this is a good thing for people with gout because they make too much uric acid. So give someone with gout allopurinol to inhibit this enzyme. And hyperuricemia is when you have too much breakdown of purines and you produce too much uric acid, and that can lead to gout. And uric acid is the soluble, excretable form of a purine. So when we break a purine down all the way, we get uric acid, and then that's what we're able to pee. If we want to salvage and recycle things rather than excrete purines and make some nucleotides once again, we could do that using two enzymes. HGPRT, so the HG is for hypoxanthine and guanine, so HGPRT can turn hypoxanthine back into IMP nucleotide, the G guanine back to GMP, and then APRT turns adenine back into AMP nucleotides. If you don't have HGPRT, you can't turn hypoxanthine back into IMP or guanine back into GMP, so you have to continue purine breakdown. And when you break down purines, you produce a lot of uric acid. So that's why someone with Leishman will have hyperuricemia. It's common in men. They self-mutilate, so they harm themselves by biting and hitting themselves, and they have retardation and dystonia, so uncoordinated movements. HDPRT is regulated, so if you have hypoxanthine, HDPRT will be able to do its job. But if you have the product of what hypoxanthine is doing, that'll inhibit it from working. Almost done. Pyrimidines, like I said, are cytosine, thiamine, uracil. Cytosines in both DNA and RNA. Thiamines in DNA, uracils in RNA. When you add a sugar to cytosine, it becomes cytidine. When you add a sugar to thiamine, it becomes thymidine. When you add a sugar to uracil, it becomes uridine. These are the pyrimidine nucleosides. When you add a phosphate to any one of these, or 2 phosphates or 3 phosphates, you get these. And since thiamine's only in DNA, you only see the D version. Uracil's only in RNA, but you see these DNA versions because they're just produced in the pathway of pyrimidine synthesis to get from U to T, but they don't function in DNA. Pyrimidine rings are also made up of other things, so I remember your cell saying, "Gah, we need more pyrimidines to make DNA." So Gah is glutamine, aspartate, and the H for bicarbonate. So carbamoyl phosphate, you could say aspartate and carbamoyl phosphate make a pyrimidine ring, or you can break them up. And just know that what each of them is giving. So aspartate's an amino acid, it'll give a nitrogen group. Carbamoyl phosphate will also give a nitrogen. And they'll both give nitrogen and carbons. Pyrimidine synthesis is pretty long. You have glutamine and bicarbonate, like we just saw, turning into carbamoyl phosphate using CPS2. We saw CPS1 in urea cycle. This is CPS2. Carbamoyl phosphate turns into carbamoyl aspartate using aspartate transcarbamylase. Dihydroorotate turns carbamoyl aspartate into dihydroorotate. Dihydroorotate dehydrogenase turns dihydroorotate into orotate. Orotate phosphoribosyl transferase turns orotate into OMP. OMP decarboxylase turns OMP into UMP. UMP, if you add a phosphate, turns to UDP. Then ribonucleotide reductase, that's that same enzyme that turns the RNA version into a DNA version. So you get these non-functional DNA versions with uracil, but they don't work. They're just used to make the thymidine nucleotides. So I remember you dump Trump. Thymidylate synthase and B9 are used to turn dump into DTMP. And then we can add a phosphate and get DTDP, add 2 phosphates and get DTTP. There's a lot happening here. So this first enzyme starts with C, this second starts with A, and this one starts with D. And this CAD gene regulates these 3 enzymes at the transcription level. So if you don't have this gene, you'll have a problem with pyrimidine synthesis. CPS2 is regulated by, so it's stimulated by ATP, so that makes sense because the A nucleotides in purine synthesis stimulate the T nucleotides in pyrimidine synthesis. And PRP will stimulate it because PRP is the, or PRPP is the template used in purine synthesis. So purine synthesis will stimulate pyrimidine synthesis because we always have A's paired with T's and G's paired with C's. UDP and UTP will inhibit this because this pathway is making uracil nucleotides. So if we have them, then we don't need to do this pathway. These two enzymes, let's start with O, that are leading to the production of UMP. They're regulated by the UMPS gene. If you have a defective UMPS gene, one of two things can happen. You can have type 1 erotic aciduria, which is where both of these enzymes don't work. If both of these enzymes don't work, then we won't be able to produce pyrimidine nucleotides. And if we can't produce pyrimidines for red blood cells, then they won't have DNA and they'll just be large red blood cells that can't replicate and divide. They'll just be big cells with no DNA, so it's megaloblastic anemia. Type 2 erotic aciduria is when only one of these enzymes doesn't work. It doesn't matter which one, it's just one of them is not functional. In order to treat this, you can give the person oral uridine, because if they don't have these enzymes, we can't get to uridine, so we need them to get it through diet then, because we need to make DNA. Our cytosine nucleotides are made from UT. So the U can make C nucleotides and T nucleotides. There's a drug, a cancer drug, 5-fluorouracil, that inhibits thymidylate synthase. If you inhibit this enzyme, you can't make the thymidine nucleotides. If you can't make thymidine nucleotides, you can't make DNA, essentially, and that's good for cancer cells because cancer cells thrive off of cell replication and replicating DNA, so that's a good thing for cancer cells. But 5-fluorouracil doesn't really know how to differentiate between the cancer cells and our body's regular cells, so we end up killing some of our healthy cells too, like the ones that divide a lot, like the ones in our hair or our scalp. So then that's why you get hair loss from cancer drugs. This reaction, you need B9 as a cofactor, so you get dihydrofolate turns into tetrahydrofolate using dihydrofolate reductase, and then tetrahydrofolate is turned into its N5-N10 methylene form for this reaction. If you inhibit dihydrofolate reductase, we can't make tetrahydrofolate, and then we won't have B9 for this reaction. This is good for cancer. Like this drug methotrexate can inhibit DHFR, and that'll do the same thing as 5-fluorouracil. It will inhibit thiamine nucleotide synthesis, and if you don't have that, you won't have DNA production. And if you don't have DNA production, you slow cancer cell proliferation, but you also kill some of our healthy cells. Cancer drugs inhibit thiamine because thiamine is the only pyrimidine that's in DNA exclusively. If cancer drugs, or if there was a drug that was targeted to inhibit cytosine or uracil, that will inhibit with RNA synthesis, and if we inhibit the synthesis of RNA, we won't be able to make proteins. And then, obviously, if we don't have proteins, we could die. Pyrimidine catabolism is pretty simple. Cytosine and uracil go through the same pathway. And thiamine is kind of on its own here. So cytosine and uracil turn into dihydrouracil. Thiamine turns into dihydrothiamine. So you see dihydro on both sides. Dihydrouracil turns into N-carbamoyl beta-alanine. Dihydrothiamine turns into N-carbamoyl beta-aminoisobutyrate. So N-carbamoyl on both sides and beta something. You just remove the N-carbamoyl and you get beta-alanine. You remove the N-carbamoyl and you get beta-aminoisobutyrate. You also have some ammonia produced that needs to go through the urea cycle to be turned into urea so that it could travel to the kidneys to be excreted. And just a side note, the salvage of purines is more important than the salvage of pyrimidines. We didn't learn about pyrimidine salvage, but it occurs. And this is probably because pyrimidines are more readily, I guess the substrates to make pyrimidines like glutamine and bicarbonate are more readily available in our bodies. So we're constantly easily able to make them. Purines take a lot more components to make, so their salvage is more important for this reason. And I think that covers everything. Hope that made sense.